lifestyle and everything have occurred. So, by knowing this evolutionary history, we'll be able to learn far more about ourselves than we could ever do by delving into ourselves alone. And as I've said, we are now in the midst of a massive sequencing effort of genomics, in which we are sequencing members of every single lineage of the living primates, as well as lots of other mammals. The rat and mouse, of course, were done rather early. The dog and horse and cow and elephant and all sorts of other mammals have also been sequenced. And this vast amount of data actually requires something very important, and that is increasingly more powerful bioinformatic tools. And I'm going to spend a little bit of time talking about what we call bioinformatics, because in my view, this is one of the futures of our field. It is the growth field of the biological sciences, computer science, and information sciences. And I would encourage all of you to explore this aspect of the field, because the lab work, while critical and important, is actually becoming easier and easier. I'll give some examples of that in a moment. And our ability to actually sequence these genomes recently has taken a really spectacular turn. And that is, we are almost finished within a year. The complete Neanderthal genome is going to be sequenced. All 3 billion bases of the Neanderthal. They've been extinct for 30,000 years. They are our closest former relative, and by looking at their genome and comparing it to ours, we're really going to be able to see some of the things that have affected us with, say, the advent of agriculture, the shifts in our diet, our health, and all of these things, and our massive population explosion and the infectious diseases that go along with that. These things wouldn't have been there for Neanderthals, so by comparing the Neanderthal genome to our own, we're going to learn even more about ourselves. So here are the genomes that are currently either finished or nearly finished. And as this has gotten cheaper and cheaper, we're going to see more and more of these done. Right now, there's a major competition going on so that people can try to get the cost of sequencing an entire genome down to $1,000. And if that happens, there could easily be a day where we all have our genome sequenced, and when you go to the doctor, he or she plugs in, I don't know what form it'll be, if it'll be a DVD or a jump drive, pull off all of your information and actually tailor your medical care. Because we know certain genes, certain alleles of certain genes are affected differently by different drugs. So that if you have to take a cholesterol-lowering drug, if you have one allele, your cholesterol will go down, but if you have another, it could go up. And so by actually knowing that in advance, by having that information in advance, our health care will be better and more efficient in the future as well. But that is going to require lots and lots of additional work. So, for my own research, we actually sequence and characterize DNA from hundreds of different species. And, believe it or not, one of our main sources of DNA from these species is their feces. So here we see gorilla feces that one of my graduate students collected. She's now a professor at Yale University. And it turns out when an animal defecates, several hundred cells are sloughed off, and we can actually tease apart and pull out those cells, the DNA in them, so that we can sequence that animal. The importance of this is that we don't have to disturb these animals. Believe me, you do not want to go up to a 400-pound gorilla and try to draw blood. That's just not a viable option. There are other animals that only come out at night. They're very hard to see. Others in large herds. And almost all of these animals are highly endangered. And in fact, and I'll make this point in a few minutes, We don't want them to be used to humans, because unfortunately humans hunt and kill many species of primates. Many species of primates are on the verge of extinction because of hunting pressure from indigenous peoples in different areas of the world. And so if they're used to humans, then a hunter can also get close to them. So in fact, we try to keep our distance. We can detect what parasites they might have. We can even figure out what these animals eat, because in their feces is not only their own DNA, but everything they have consumed. So we can actually extract the complete set of DNA out of the species, and using very targeted probes, figure out which plants, which insects, maybe which animals that they've eaten in the last several days. So we can actually work out their diet. We can work out paternity. So when you see a large male silverback gorilla with a group of females and all of the offspring, we can actually test to see if he indeed is the father of all of those offspring. So we use this in behavioral studies, relationship studies, as I said, diet studies. We even have a pilot project going in South America in which we've actually DNA fingerprinted individual fig trees in the forest, and when we extract the DNA out of their feces, we can tell exactly which tree an animal actually traveled to and ate from. So we can actually track and follow these animals through tens of kilometers of the rainforest based on what they've eaten, where, and then what they've left behind after that. So it's very important. I once gave a talk in which, to paraphrase the old NAACP phrase, I said, waste is a terrible thing to mind, because we work with feces and waste almost daily in the lab. This small amount here has enough DNA in it for me to completely genotype and characterize an animal. We've actually done census experiments on gorilla populations in Nigeria of animals that we have never seen. But by collecting their nightly nest feces, week after week, genotyping it, determining, just like in CSI, getting an actual DNA fingerprint of every individual till we find no new individuals, we can determine exactly how many individuals live in that troop, how many are male, how many are female, how many have the same father, the same mother, and even work out their relationships without even seeing these animals. This is such a revolutionary technique. We can work on mitochondrial DNA, which one of my students will talk about after my talk here. We can get at other parts of the genome and do this. As I said, scientists in Germany and in Connecticut are on the verge of publishing the complete Neanderthal genome. It's using many of the same techniques. In this case, getting the DNA out of the few intact cells left behind in bones. So in my lab, we can extract DNA from feces, from hair, from saliva, from bone. In fact, we almost never get whole animal tissue or blood or anything that would be nice. That's really easy to work with. But using our techniques, we can actually zoom in on really small, tiny, minuscule